Let us see the active site. What do you mean by active site? Active site of an enzyme is comparatively small, comprises comparatively very small region. খুব ছোট্ট অংশ অকুপাই করে থাকে গোটা এনজাইমের তুলনায়, যেখানে সাবস্ট্রেটকে বাইন্ড করার একটা জায়গা থাকে, তাই না? সাবস্ট্রেটকে বাইন্ড করার বা হোল্ড করার জায়গা থাকে। সেই অ্যাক্টিভ সাইটকে আমরা এই কতগুলো দিতে পারি, কি বলে ফিচার্স বা প্রপার্টিস অফ অ্যাক্টিভ সাইট যদি বলো, সেগুলো কি না, সাইজে খুব ছোট হবে, এখানে কিছু স্পেশাল রেসিডিউ থাকবে। কি রেসিডিউ থাকবে? দেখা যায় সাধারণত এখানে হাইড্রোফিলিক রেসিডিউস থাকে, মানে চার্জ রেসিডিউ। ইউজুয়ালি কি হয়, আমাদের ব্লাড বা সিরাম যাই বলো বা প্লাজমা বা সাইটোসল, এগুলি মোস্টলি ওয়াটার। সো the proteins are folded in such a manner so that the hydrophilic parts are always jutted outwardly, মানে বাইরের দিকে থাকে, প্রোটিনের মন্ডটার বাইরের দিকে থাকে হাইড্রোফিলিক গুলো, যেমন গ্লুটামেট, অ্যাসপার্টেট, যেগুলো চার্জ থাকে। কিন্তু দেখা যায় যে অন দ্য কন্ট্রারি অ্যাক্টিভ সাইটে কিছু কিছু চার্জ পার্টিকেল থাকে যেগুলি ওয়েল ইনসাইড দি স্ট্রাকচার অফ দি প্রোটিন। তার মানে ওই স্ট্রাকচারগুলো, দ্যাট মিনস দোজ চার্জড রেসিডিউস অর হাইড্রোফিলিক রেসিডিউস হ্যাভ সামথিং টু ডু, some function to perform. What is that function? Mostly they hold the substrate tightly in a well covered area. 99% of reactions require an atmosphere where only substrate can occupy the active site, at the most some co-substrate, but there is no place for water inside the enzyme structure. Why? Because water inside is not invited, not a guest, invited guest. It's always repelled outside. So the active site has two parts, binding site and catalytic site. So which one is bigger, binding site or catalytic site? Obviously binding site. Binding site means the seat in the aeroplane flight, is a binding site. Active site, the seat belt, correct? Active site is the seat belt, correct? So the small region which holds the substrate, a catalytic site is a seat belt, the small region which holds the substrate in position. How do we determine the active site? By these three methods, separately or in combination. Use of specific inhibitor, just find out the specific inhibitor, let it bind with the substrate enzyme, and then take a photograph either by crystallography or some by other means. First you have to freeze them, then use the X-ray crystallography to take the photograph where actually the inhibitor is sitting inside the enzyme. Follow? So that region we can explore further in the enzyme to find out the actual structure or structural feature of the active site. So first we need to find one inhibitor, then we will let it bind to the enzyme, and then we will freeze it so that they do not move, and then we will take a photograph to find out where is actually it is located, which position of the enzyme it has occupied. Types of enzyme specificity: absolute specificity, group specificity, bond or linkage specificity, stereo specificity and geometric specificity. All these we should remember at least with one example. Absolute specificity. This is example of absolute specificity, correct? Glucokinase, fructokinase, example of absolute specificity. They act only on one type of substrate and nothing else. Group specificity. Hexokinase. Why? Because these two both are hexose. Both fructose and glucose can be acted upon by hexokinase. Both will produce corresponding 6-phosphate product. Third, this will produce what? And this will produce, but this will produce correct? Corresponding sugar 6-phosphate. Bond or linkage specificity. Chymotrypsin, they will cleave the peptide linkage where this group is donated by aromatic amino acid. Correct? Aromatic amino acid. So this is a correct? Stereospecificity. Yeah. D-amylase oxidase. So these L and D are stereoisoform. They are stereoisoform, but they have two separate enzymes. Both the enzymes will catalyze same type of reaction, that is oxidative deamination. But for L amino acids there is a separate enzyme, and D amino acid there is a separate enzyme. Geometric specificity. One very good example is this one. Another example is also there. This one will be difficult for you to do you remember what do you mean by geometric isomerism? Cis trans, correct? Achha. So, have you heard of this pathway? Huh? Beta oxidation. It is a pathway which oxidizes fatty acid, correct? The beta oxidation pathway not only oxidizes the saturated fatty acid, but also it oxidizes unsaturated ones too, correct? The unsaturated fatty acid can be of two types, either of cis configuration or of trans configuration. So these enzymes are mostly they mostly act on trans form, not cis form. If there is a cis form, then they will convert it into trans and then they will follow the oxidation step. Correct? So this is an example where geometric specificity can we mention? Now, this is something bit difficult. Now we are actually plunging into the catalysis process. The catalysis process is not as simple as like the enzyme comes and sees the substrate and love happens and then reaction. No. It requires proper adjustment, sitting arrangement and binding to a particular catalytic site. How does it happen? It is like standstill, like the hero and heroine looking at each other's eyes? Huh? Like that? No. Then how is it like that? I mean, how it is like? It is like a motion, always, perpetual motion. Means enzyme and substrate, they are just colliding. They will go on colliding with each other for infinity, for time infinity, unless the energy dissipated by the collision exceeds the activation energy required to convert substrate to product. Correct? That means they will continuously collide. At some moment of time, when the effective kinetic energy of these colliding molecules will increase. How they will increase? By the temperature. If we increase the temperature of the system, then they will collide with greater velocity, half mv square will be increased, and this kinetic energy will be converted into energy dissipated by the collision, which should be more than that of the activation energy. What is this activation energy like? The y-axis shows the quantity, the amount of energy, quantification, and this is time, correct? Now, this particular energy is called so there is a wheel here. If you push the wheel uphill, bring it here, then it has equal tendency to roll down on either direction. আমি যদি একটা চাকাকে টেনে হিঁচড়ে উপরে নিয়ে যাই, তাহলে এই পয়েন্টে চাকাটা এদিকেও যেতে পারে, এদিকেও যেতে পারে। সাবস্ট্রেটের এরকম স্টেটকে বলা হয় ট্রানজিশন স্টেট। ট্রানজিশন স্টেট কাকে বলা হচ্ছে? The transition state is a state of substrate where substrate can go to the formation of either substrate itself back or the product. It can go in any direction. How this will happen? By the laws of nature, it will happen if you wait for thousands of years. It will happen naturally. Or you have to add some enzyme. What enzyme will do here? Enzyme will as if form a tunnel through the, as if it will make a tunnel through the hill. So now the wheel can pass through this. So now this will become delta A, right? So the activation energy is lowered. So enzyme will lower the activation energy. How it will lower the activation energy? These 6 methods. Either one of these, 2 of these, 3 of these, or all of these can be used together to bring down the activation energy of the substrate for the conversion towards a product. Right? So, substrate to product, this particular diagram can be explained under the heading of tunnel theory. Correct? So, mechanism of enzyme catalysis means this, how to reduce the activation energy. Is it clear? Previous class we discussed how enzymes act, the 3 theories, how they actually bind with the substrate. This class we are discussing the enzymatic catalysis process. 2 things are different. 2 things are different. Correct? This is a process of catalysis by which they will actually reduce the activation energy. So the first one comes general acid-base catalysis. It is comprised of 2 theories, 2 components, general acid catalysis and general base catalysis. Together they form acid-base catalysis. Acid-base catalysis means what? One of the component will give away H plus ion, another component will take away H plus ion. The one which will give away H plus ion will be acid, the one which will accept it will become base. So if it happens so, then it is called acid-base catalysis. Let's go through it. You need not to remember all this in detail. Hello? Simple paper. One component will donate H plus ion, it will be called as general acid, and another component will take up the H plus ion, it will be called as general base. If these 2 are working together, that catalysis is called acid-base catalysis. This is a mechanism. Done? Examples of different amino acids. Which amino acid can act as donor, which amino acid can act as acceptor? Donor means what? See? Histidine. This is a basic amino acid. But this is what? You remember that Arrhenius theory, Arrhenius acid and Lewis acid, Arrhenius base, Lewis base. So anyone who gives away H plus ion will be Arrhenius acid. But histidine is a Lewis base, imidazole ring is a Lewis base because it has got NH3 plus. Correct? So I am not going into detail. Electrostatic catalysis. Electrostatic means static electricity. Why do you find static electricity most prominent? The area or the place where there is no water. Why? Water has highest dielectric constant. Correct? It has highest resistance, pure water, for electricity. So one positively charged and one negatively charged component will cling together if there is no water. So the first and foremost criteria for electrostatic catalysis is that internal milieu of the enzyme substrate interface. Internal milieu of the enzyme substrate interface should be devoid of any type of water. Correct? Then only they can act. One will be positively charged, another will be negatively charged. Positive charge means what? Lysine, arginine, histidine. Negative charge means aspartate, glutamate. Covalent catalysis. Now, as I mentioned, the reaction can only take place when enzyme and substrate come in contact with each other. It is not telepathy. It cannot be done over the telephone. You have to come and touch and hold and hug. Then only it will happen, otherwise it will never. Correct? But hugging means what? You have to hold it tightly. How tightly you can hold will decide how faster the reaction can take place. Right? For holding together all these methods, it might be through electrostatic mechanism. That means an what? কি নিয়ে বলে? অদৃশ্য কি বলে? Invisible attraction. Electrostatic attraction means invisible attraction. Means there is an attraction but which is invisible. It's called electrostatic attraction. But when the attraction is visible, like during the Hindu marriage, তাই না? ওই যে ধুতি আর শাড়ি বেঁধে দেয়, when the attraction is visible, then it becomes covalent bond. Now the bond is formed, you cannot go anywhere. তাই না? Now you can ask me that which one is more stronger, the invisible electrostatic bond or the visible covalent bond? Now the fallacy is, me being Hindu, if I say the covalent bond is stronger, you will say I am doing. But there is a fallacy, paradox in chemistry. The paradox is, alone covalent bond is no doubt stronger than electrostatic interaction. There is no such word called electrostatic bond. It is always electrostatic interaction. But as a matter of fact, electrostatic interaction can never be a singular. I'll give you an example. What is this? A cube. The structure of the crystal of NaCl. They exist in this form. Every sodium is surrounded by 6 chloride and every chloride is surrounded by 6 sodium. So electrostatic interaction can never be alone. So together in a small region, small area. How small? What is the bond length of covalent bond? What is the smallest length of covalent bond? One point? Bolo? Ready? The smallest? And largest? Right? You cannot forget chemistry like that. Huh? 1.4 angstrom. তাহলে যা কিছু বন্ড হবে এর মধ্যেই থাকবে। কিন্তু ইলেকট্রোস্ট্যাটিক ইন্টারঅ্যাকশন কত হবে? In a range of It has to come very close. It has to come very close. They don't need to be very strong. It is stronger than covalent bond 1,000 times, but in a very close situation. Right? So the covalent bond, covalent catalysis actually provides the immobilization of the substrate, so that substrate cannot move. Substrate can move in two ways. One is like this, transverse distance covered by the substrate, another is torsional distance. Correct? They can rotate in the same axis. Both the movements should be stopped in order to reaction to take place. Clear? Covalent bond will do the same, like this. Electrostatic bond will do the same, like this. Acid base, same, like this. Electrostatic interaction will hold. One will be positive charge, another will be negative charge, and they will hold together. Fine? This one you can write, this often comes in the examination. Role of metallion in enzymatic catalysis. Role of metallion in enzymatic catalysis. What is that? Metallion can participate in two ways in enzymatic catalysis. One, they can be part of the structural component of the enzyme itself, or they can just participate from outside as a cofactor. Correct? Like magnesium in hexokinase, zinc in carbonic anhydrase, is a part of the structure, whereas magnesium is like a cofactor. Followed? So the compounds which contains metallion as a part of their structure, they are called metalloenzymes, like catalase, SOD, xanthine oxidase. All these are metalloenzymes. And the enzymes which require metallion for their catalytic activity are called metal activating enzymes. They require metallion for their catalysis purpose, not for the structural component. For them, mostly like sodium, potassium, magnesium, calcium. Say for example, calcium is required for phosphorylase activity. Magnesium is required for any kinase activity. Correct? Sodium, potassium is required for pyruvate kinase activity. Correct? Like that. Proximity and orientation effect. Now I would like to have two volunteers. Stand here. Come. Now don't move from your place, okay? Now shake hand. Shake hand. Don't move. Try. हाँ? नहीं होगा? क्यों? हाँ? क्या है? दूर है? माने? Which word will you use there? Proximity or orientation? So the reaction to take place, substrate should be near. नजदीक आ जाओ, पास आ जाओ. Now they are very much close. Move this side. Now second. Shake hand with your right hand. Is shaking hand? Are they shaking hands in normal way? Then they are very close. Now you can shake hand formally and go. Correct? See? So simple things. Molecules can never react unless they are properly placed. They should not be properly placed, they should be properly oriented. Otherwise, alone proximity won't do, alone orientation won't do. Correct? Can you give one example of this? Anyone who is very good in chemistry, he can give the example. Have you heard of nucleophilic attack? How it happens? They attack from back. You can never see who is coming. They attack from back. All SN1 reaction, right? SN2. SN2. Very good. So, sit down. So, these are the required points. The last one. Now, if you remember, if you can recall the last theory of how enzymes act, what did I tell you? It is not the substrate which is complementary to the active site of the enzyme, but it is a transition state of the substrate which is complementary to the transition state of the enzyme. Neither enzyme nor substrate are having complementarity in their interface, in their native structure. They have to move on to top of the mountain so that they can have their transition state. After achieving the transition state, what they achieve? They actually bring about subtle, small amount of changes in their binding interface so that they can fit much better compared to their native state. Native state means as if the shape with which they were born. You know? So, transition state, stabilization stage, like it is a conformational distortion. What is that? Have you seen a bow and arrow? What happens in bow and arrow? How does it function? What changes take place? Strain. When we release the strain, it comes to a normal shape and that energy throws the arrow to the target. So, the molecule is always powerfully bent and fit into a small place in the active site, and the molecule will always try to bring back its own shape, but it cannot. But with this, during this try, it will dissipate some energy, and that energy is actually required to reduce the activation energy. That is why enzyme catalysis can take place at very moderate temperature, like body temperature 37 degrees Celsius, 1 atmospheric pressure. It does not require very high atmospheric pressure. Why? Because the enzyme itself will bend it and push the substrate to the active site. All you require is a slight amount of heat energy which is provided by our body. If it is taking place outside the body, then we have to rely on the atmospheric temperature. That is why it takes less time to curdle in summer, but more time to curdle in winter. Less temperature, less speed, so it takes more time to form curd. So try to remember the metal ions and the enzymes in which they are found or their functions require. Magnesium, manganese, molybdenum, Fe2 plus, Fe3 plus, copper, potassium, selenium, nickel, zinc. Try to remember the odd ones more, because that is a target for examination. Very few will ask you, tell him just take any enzyme which require copper or iron, but many will ask you tell one enzyme which require nickel, molybdenum, zinc. Apart from these enzymes, there are many enzymes which require the same thing. Now see here, all are metals. There is a non-metal very commonly found. What? Chloride. Chloride. Can you name one enzyme which required chloride for activity? Correct. Amylase. Amylase requires chloride for its activity. Very good. Now, one of the long question. Factors affecting enzyme activity. There are many other, in addition to these, light, sound, but we will remember at least these 6. Sufficient for us to pass and get good marks. Enough. Correct? Nowadays, getting highest marks has no value. Yes. If you get 100 out of 100 and someone gets 50 out of 100, both of you will be passed. Because God knows when they start next again. Gold medal with next failure, no doctor. Just pass with next pass, doctor. So don't try to get full marks. Just pass. Just lower your ambition or aspiration a little bit and think practically. So what are the factors on which enzymatic activity depends? Number 1, more hands, more work, enzyme concentration. Number 2, temperature. I told you, what is that? Effective kinetic energy depends on temperature of the perpetuating molecules. pH regulates the ionic state of the residues present in the catalytic site. If pH is increased, it will be negatively charged. If pH is decreased, it will be positively charged. Substrate concentration, difficult. With going substrate, it will increase, then it will become standstill. Inhibitors, any inhibitors, there are many types of inhibition, may interfere with the substrate for binding to the active site, or in different other ways will reduce the rate of production. Cofactors, availability of cofactors will definitely bring about changes in the production. This reaction is easy to understand. The free enzyme reacts with free substrate, forms an enzyme substrate complex, that is transition state, right? Then it has equal tendency to go to either direction. It can form product, it can form substrate as well. So this reaction, rate of the reaction is rate of the reaction. If we consider from substrate, rate of reaction if we consider with product, only this much, nothing else. Only this much. D minus density means what? Plus dBD means what? Tell me what, that's all. Minus density means substrate concentration is decreasing with time. dBD means product concentration is increasing with time. Means the graph for substrate will be, graph for product will be, right? है ना? It is a minus dsdt. Right? So let's remember this. We will come. We will require this later. We will stop here, na? I think before starting today, only 2 minutes are there. We will stop. Next class we will see. Huh?